You know, in the world of genetics, we are practically drowning in data. But what if a huge chunk of that data is, well, just wrong? Today, we're going to look at a super smart new tool designed to be a kind of digital detective, cleaning up our massive genetic databases to make sure science is built on solid ground. So first, let's meet our main character, the microRNA, or miRNA. The best way to think about these is like tiny little dimmer switches inside our cells. They don't just flip genes on or off, they fine-tune their activity. And this regulation, it's absolutely crucial for, well, pretty much everything, from how we develop, to our metabolism, to our body's defense systems. They're essential for life in both plants and animals. So because these little molecules are so incredibly important, you can see why scientists are so desperate to find them. But the problem is, it's like trying to find one very specific needle in a mountain of haystacks. And even worse, what if a lot of the needles they think they found so far aren't even needles at all? And look, this isn't just some abstract theoretical problem. We're talking about a massive case of mistaken identity happening right inside our genetic code, and the scale of it is pretty staggering. Okay, to give you a sense of the scale here, just look at this number. The main database for these molecules, it's called miRBase, has over 48,000 mature microRNA sequences cataloged across hundreds of species. I mean, that's an enormous library of biological information. So the big question is, how many of those 48,000 entries are actually imposters? How many are just, genetic noise or some other type of molecule that got completely misidentified along the way? So, why is this such a big deal? Well, these false positives create what you could call dirty data, and that seriously holds back research. Imagine you're a scientist spending years, maybe millions of dollars, studying a molecule you think is the key to curing a disease, only to find out it was a case of mistaken identity all along. It doesn't just waste time and money, it can send entire fields of science down the completely wrong path. Okay, but here comes the hero of our story. It's a new tool that's designed to tackle this data pollution problem head-on. Let's meet miRScore. Probably the best way to get your head around miRScore is to think of it like a bouncer at a really exclusive genetic club. It just stands at the door of the database, and it checks every single candidate that tries to get in. If a molecule doesn't meet the super strict criteria, nope, it gets turned away. Only the real, high-confidence microRNAs get to make it past that velvet rope. And what you're seeing here shows a really important detail about how miRScore works. It's not a discovery tool itself. It doesn't go out and find new microRNAs from scratch. Instead, it's a validation step. It comes in after other programs think they've found something, and it acts as the final quality check before that discovery gets submitted to a public database. It's this standardized checkpoint that the whole field has frankly been missing. So, how does this genetic bouncer actually do its job? Well, its method is surprisingly simple but really powerful. It uses a two-point check to sort the real from the fake. It really just boils down to two fundamental questions. First, there's a structural check. Does the molecule actually fold into the very specific hairpin shape that all real microRNAs are supposed to have? And second, there's an expression check. When scientists look at the genetic data from actual cells, is there any real evidence this molecule is actually being made? In other words, does it even show up? And the secret sauce here is specificity. For the structure, it's looking for that perfect hairpin shape with a tiny, precise two-letter genetic overhang right at the end. For expression, it's not enough for the molecule to just be there. It has to show up at least 10 times in the data, what scientists call reads. And here's the most important part. Over 75% of the genetic signal from that spot has to map exactly to the microRNA. This precision check makes sure it's a real signal, not just some random background noise. And this, right here, shows you the difference perfectly. On the left, you've got a microRNA that passes the test. See how clean and tight that hairpin structure is? Now look at the one on the right. That one fails. The structure is a bit messier, sure, but the real problem is what the colors are showing us. The expression data just doesn't line up neatly with the structure, and that's what causes it to fail that critical precision test. Okay, so the theory sounds great, right? But how does miRScore actually perform in the real world? Well, the researchers put it to the ultimate test against some massive existing databases. The researchers didn't just build this cool tool and call it a day. No, they unleashed it on the biggest, most widely used databases out there, like miRBase, which we already talked about. They ran it on data from humans, from mice, and from several major plant species too. And wow, the results were pretty striking. In these charts you're looking at, the green part of each bar shows the entries that passed miRScore's test. Everything else failed, either for structural reasons, expression reasons, or both. As you can see pretty clearly, miRScore flagged a huge number of existing entries, which suggests that a lot of our official genetic library just doesn't meet these strict quality standards. Let's just zoom in for a second on the human data from miRBase, which is what most medical research, for diseases like cancer, relies on. The results here are just staggering. Only 21%, just over a fifth, of the entries actually passed miRScore's validation. That means almost 80% failed for one reason or another. I mean, think about that. The very map we're using to navigate and find cures, it might be seriously flawed. And here's another really cool thing miRScore can do. It can act like a referee for other scientific tools. Researchers used it to judge 3 different programs that are used to discover microRNAs, and the results were just night and day. As this chart shows, one tool called ShortStalk had a perfect 100% pass rate, but another popular tool, it had a pass rate of only 21%. This means miRScore can actually help scientists pick the right tools to generate high-quality data right from the very start. So what's the big takeaway here? It really comes down to a simple but incredibly profound idea. Better data leads to better science. At the end of the day, every single great scientific discovery is built on a foundation of solid, reliable data. And if that foundation is shaky, if it's full of false positives and dirty data, then the whole structure is at risk of collapsing. By cleaning up our databases, tools like miRScore are making sure that future research is being built on solid ground. And that's going to help us accelerate real discoveries and avoid a lot of costly dead ends. And I think the researchers behind it said it best. They state that the goal is to improve the accuracy of new submissions, while also giving us a way to reevaluate the thousands of entries that already exist. It's really a tool for raising the entire bar for data quality, both for the future and for the past. And that kind of leaves us with this one big question to chew on. We live in an age where biological data is growing exponentially. It's just impossible for humans to manually check everything. So as we move forward, how are smart automated validation tools like miRScore going to not just clean up our data, but fundamentally shape the entire future of scientific discovery itself?